Endoplasmic reticulum, and this session is going to focus on rough endoplasmic reticulum. So, as mentioned earlier, endoplasmic reticulum is an integral part of the endomembrane network, and rough endoplasmic reticulum, in fact, is a starting point of the transport of molecules or transport of proteins to various destinations in the endomembrane system. So let us look at the learning outcomes of this session. The endoplasmic reticulum is of two types, smooth and rough. The membranes of which are continuous, and you would find that they share a lot of proteins. So there are certain functionalities in both smooth endoplasmic reticulum and rough endoplasmic reticulum that are same, but the major difference is in the fact that the rough endoplasmic reticulum has ribosomes on its surface, and hence its major functioning is on synthesis of proteins and also ensuring that the proteins formed are properly folded and functional. But the point over here that has to be kept in mind is that ribosomes are also free. That is, they are present free in the cytosol. So not all proteins are formed on the rough endoplasmic reticulum. It is especially those proteins that are secreted by cells or those that are targeted to a particular destination, say for example the plasma membrane, or say for example the lysosomes, say for example the Golgi, and so on and so forth. The rough endoplasmic reticulum initiates glycosylation of proteins, so it is involved in what is called as post-translational modification, glycosylation being one major post-translational modification, and proper folding of the proteins. This is something very important that the rough endoplasmic reticulum takes care of. Moreover, membrane-bound proteins too are synthesized in the rough endoplasmic reticulum. As part of the endomembrane network, it plays an active role in transport of proteins to and fro from different organelles. So, when you look at the endoplasmic reticulum, we all know that it originates from the outer nuclear membrane of the nuclear membrane, and then it is a continuous system with the rough endoplasmic reticulum and the smooth endoplasmic reticulum sharing common membrane, continuous membrane structures, but within them again, certain endoplasmic reticulum are as flattened sac, and they're called as cisternae, and these flattened sacs can be studded with ribosomes, and they then are what is the rough endoplasmic reticulum, while certain cisternae get evolved into tubular interconnected curved structures, and these are not studded with ribosomes, and they form what is called as the smooth endoplasmic reticulum. And in this session we are going to focus on the rough endoplasmic reticulum. Now, one of the major functions of the rough endoplasmic reticulum is in protein synthesis, as ribosomes are found bound to its surface. Here, there are two questions. Uh, Before we go to the two questions, we need to know that the ribosomes that are exported from the nucleus as small subunits and large subunits can assemble into complete ribosomes when they have to carry out translation. So the cytosol contains all these large and small subunits of the ribosomes, and they can assemble to translate mRNA into proteins as free ribosomes. And mainly what has been observed is that all proteins that are required in the cytoplasm, all proteins that are transported to mitochondria or to chloroplasts, etc., are formed in the cytosol on ribosomes which are free, not bound to the endoplasmic reticulum. So therefore, the question arises is that how and why do the ribosomes go and bind to the surface of the rough endoplasmic reticulum? And also the question that comes up secondly is that which are the proteins, or how does the rough endoplasmic reticulum know which are the proteins that it has to synthesize and take forward to different destinations? So therefore, this answer was given by, was actually proposed, or rather, this was proposed by a scientist called uh, Günter Blobel, and he kind of said that the protein themselves have a signal uh, which allows the protein to be synthesized on the rough endoplasmic reticulum, and the hypothesis pertaining to this is what is called as a signal hypothesis. Now, please do understand that the proteins that are synthesized by the ribosomes are those proteins that are to be secreted by the cell. So, the secretory pathway originates or begins with the rough endoplasmic reticulum. It synthesizes those proteins that are integral membrane proteins. So, we know that membrane proteins are of two types, peripheral and integral. So, all the integral membrane proteins are definitely formed by the rough endoplasmic reticulum. And also, it gives rise to soluble proteins, that is not membrane bound, but soluble proteins. But these proteins are generally those that would reside in the endoplasmic reticulum itself or go to the Golgi apparatus, vesicles, lysosomes, endosomes, etc. And please note that all of these are organelles that are part of the endomembrane system. So these are the proteins that are going to be formed by the rough endoplasmic reticulum. Now this that you observe over here, so we will come back to the signal hypothesis in a while, but before going to that, let us look at the secretory pathway itself. So you can see that this is the rough endoplasmic reticulum where the ribosome comes and gets studied to form the protein. The protein is packaged and it is transported as vesicles to the Golgi. In the Golgi, the proteins are processed, and having got processed, some of the proteins are continuously secreted out as the vesicles go and bind to the plasma membrane and get exocytosed. So that secretion, where the secretion of the proteins or the molecules are continuous, those are called as constitutive secretion. But in some cases, what can happen is that you have the proteins being processed, made ready, but then they are stored in the vesicles in large amounts. And only when there is a cue or there is a signal, then you have these vesicles getting exocytosed through the membrane and releasing the proteins. So only when required, they are released. So the best examples for this is insulin. So insulin that is produced by the beta cells of the islets of Langerhans of pancreas are already formed within the beta cells and stored in vesicles. But when there is a cue, when the blood sugar goes down, then you have, when the blood sugar goes high, then you have a signal that is going to exocytose the stored insulin present inside and it is released outside. So therefore, you can either have regulated secretion or you can have constitutive secretion, but look at the fact that there is a transport of proteins all through from the endoplasmic reticulum to the plasma membrane, and there are many proteins which are also from the plasma membrane taken in through endocytosis and they are moving to the endoplasmic reticulum. So you have what is called as anterograde movement of proteins taking place and retrograde movement of proteins taking place. So this anterograde and retrograde movements is what is called as vesicular trafficking, which is a very important fact of the cell, and ER is part of this particular vesicular trafficking system. So as mentioned to you, what decides which proteins are to be synthesized in the RER? So we looked at the fact that there is something called signal hypothesis. So let us understand what is signal hypothesis. So what has been observed is that those proteins that have to be synthesized in the ER are first actually synthesized by the ribosome as a free ribosome. So you can see how a free ribosome is bound to the mRNA and it has already started translating using that mRNA to form the polypeptide. Now, This polypeptide, as it is being synthesized at its end terminus, there is a sequence which is called as a signal sequence. Now, this sequence of amino acids is generally conserved. And what is interesting to note is that this signal sequence generally has about 11 to 15 or 11 to 14 hydrophobic amino acids or neutral amino acids, and this stretch of or this sequence of amino acids that is present on the N terminus of the polypeptide is the one that decides that this protein has to go to the endoplasmic reticulum. Those proteins that do not have the signal sequence do not go to the endoplasmic reticulum. Now, this is very similar to, the nuclear localizing signals that are present on proteins that have to be imported into the nucleus. So, only those proteins that have the nuclear localizing signal are taken up by the nucleus, right? Similarly, in this case, only those proteins that have the signal peptide are the ones that are going to go to the endoplasmic reticulum. So the third question that we ask over here is, now that the signal peptide has been formed, so you must see that the protein is still being formed. It is not completely translated. It is just begun to get translated. And you already have the N terminus exposed, and this N terminus has the signal peptide. So once the signal peptide is formed, then how is it that this entire ribosome moves to the endoplasmic reticulum? So the question is answered by the fact that this signal sequence or this signal peptide is recognized by a protein. In fact, it's not a pure protein, it's a ribonucleoprotein, and that is called as signal recognition particle. So the signal recognition particle has RNA and it has protein. And as a combination of the two, it is forming what is called as a ribonucleoprotein. Because it has the RNA of the signal recognition particle is able to bind to the RNA that is rRNA that is present in the small ribosome. Plus, the signal recognition particle can also bind specifically to the signal peptide. So therefore, our understanding now is that the signal recognition particle is now a protein, is now a ribonucleoprotein that is able to recognize the signal peptide. So, effectively, you would have the signal recognition particle binding in such a way that it binds to the signal peptide, but it also binds to the ribosome. Now, as it binds to the ribosome, and you can see how it is kind of ensconcing the polypeptide that is being formed. This association of the SRP with the ribosome ensures that the ribosome stops translating. So all this while it has been translating, but the moment SRP comes and binds to the signal peptide and to the ribosome, now the ribosome pauses in its activity of translation. So it does not carry out translation any longer. It has stopped, it has paused. Okay. So right now the ribosome is in the pause mode for protein synthesis. So therefore, as mentioned over here, when it binds to the ribosome, it can halt translation. What can halt? The signal recognition particle. One interesting fact that you should keep in mind over here is that this signal recognition particle is able to bind to GTP. And when you have the SRP as SRP GTP, then it can bind to both the signal peptide and to the ribosome. So this fact is again important, that SRP GTP is the form, because as SRP GTP, it has a conformation that makes it have an affinity to bind to the signal peptide as well as to the ribosome. Now the next question comes is that this now as a complete complex. So you can see that it is a complex which contains mRNA, it contains the ribosome, it contains the signal recognition particle, and it contains the newly synthesized polypeptide. So this complex now is to move to the ER. So the question is, how is it able to now go and bind to the endoplasmic reticulum? So let us look at the endoplasmic reticulum itself. So you can see that this is the endoplasmic reticulum, this part is the membrane, and this is the ER lumen. So on the membrane of the endoplasmic reticulum is present what is called as the signal recognition particle receptor. This too has different conformations when it is bound to GTP and when it is bound to GDP. So that also is a kind of a regulatory thing. So both SRP and SRP receptor are able to bind to GTP and GDP, and depending on whether it is bound to GTP or whether it is bound to GDP, their conformations will be different. So just beside the signal recognition particle receptor is present another protein complex, again another transmembrane protein, which is called as the translocon. And interestingly, this translocon has a pore, but the pore is plugged by a helix which contains a stretch of amino acids that is hydrophobic. So this helix is kind of forming a plug, okay, and to this SRP receptor comes and binds the SRP, as you can note over here. So the second step is basically that the SRP comes and binds to the SRP receptor, and this plug, as you can see this is the plug, this is the pore ring, and this plug over here is keeping the pore ring closed, so nothing can really enter, okay, unless the pore ring opens up or the plug is shifted. So this is something that is important. You have the translocon, but nothing is going to translocate unless the plug is closed. So what happens next? So what happens next is that the SRP, on binding to the SRP receptor, immediately has its GTPase activity such that the SRP GTP becomes SRP GDP, and in the SRP GDP form, it doesn't have affinity to any longer bind to the ribosome and to the signal peptide, so it leaves. So you can immediately understand that this is a kind of binary switch. When SRP is bound to GTP, it has affinity to bind to the signal peptide. But when it gets converted to SRP GDP, it releases itself from the ribosome and from the signal peptide and goes away. And why not? Because it has done its job. What was its job? Its job was to bring a translating ribosome to get studded on the surface of the endoplasmic reticulum right on the translocon. So you have the ribosome lodging itself on the translocon in such a way that the signal peptide is pushed towards the plug. Okay, now since it is hydrophobic in nature, and because the ribosome is now bound to the translocon, there is a conformational change in the translocon, releasing the plug, and now the signal peptide can bind to this pore region. But the pore opens, and so what happens is that although the signal peptide can remain bound over here, the remaining polypeptide that is formed will now get translated but translocated as well. So you can say that translation and translocation is happening together. So it is translocatory. Okay. Now, interestingly, what has been observed is that the endoplasmic reticulum membrane has an enzyme that is called as signal peptidase. So that signal peptidase will cleave off the signal peptide. Okay, so signal peptide will either remain bound to the membrane or it will be released into the lumen, and then that can get degraded because the job of the signal peptide is also done. What was the job of the signal peptide? To ensure that it binds to the translocon, it opens up the pore, okay, and now the protein is being translated, but the protein that is translated is entering the ER lumen. Now, once it enters the ER lumen, the protein that is entering the lumen will have to fold properly. So another function of the endoplasmic reticulum is that the endoplasmic reticulum ensures that a protein is folded properly. So that is one of its major functions. So as you can see over here, as the protein is translocated inside, the protein is folded properly, okay? And this folding of the protein can take place spontaneously on its own. But if it is not folded properly, then there are certain proteins called molecular chaperones. So these molecular chaperones are able to bind to those proteins that have not folded properly, and by binding to the misfolded protein, they can unfold the protein and allow the protein to be folded again properly. So the molecular chaperones are going to help the protein fold properly. But despite its efforts, if the protein is not folded properly, then what has been observed is that those unfolded or misfolded proteins, okay, they are bound to by certain factors called the endoplasmic reticulum associated factors, and these factors would guide those unfolded proteins out into the cytoplasm. So you can see from the lumen of the ER, it is going into the cytoplasm. When it is going into the cytoplasm, it gets ubiquitinated. So there are small peptides called ubiquitin that come and bind to the polypeptide that is being released from the ER. Why is it being released from the ER? Because it was not folded properly. If it is folded properly, it will remain inside the ER lumen, okay? And then we will look at what happens to that protein. So as the protein gets ubiquitinated, a ubiquitinated protein is generally a tag that this protein needs to be degraded. So the ubiquitin guides that protein to something that is called as proteasomes. The proteasomes are again molecular complexes that have a lid. Through the lid, the protein is going in, the ubiquitin is released. Inside the proteasome, the protein is degraded to amino acids or smaller peptides and released into the cytoplasm. So, in fact, what is observed is that these proteins are recycled. Because they did not fall properly, they're again recycled, and the cytoplasm has again amino acid pool that can be used for synthesizing a new protein. So that is something that is associated with endoplasmic reticulum. Those proteins that have folded properly, they can either remain in the ER to function in the ER, or they can be exocytosed out through a vesicle and can be carried to different destinations. So here it is important to note that if the protein has to stay within the endoplasmic reticulum, then there has to be a signal again. The protein or the endoplasmic reticulum has to know that this is a protein that it has to keep for itself. The rest it can send away to different destinations, but that protein that is required for functioning within the endoplasmic reticulum, or that protein that has to become part of its membrane, has to remain in the ER. So the question is, how does the endoplasmic reticulum know that the particular protein that has been synthesized has to be retained in the ER? So the protein that has to be retained in the ER has something called ER retention signal. Again, a sequence of amino acid. Now, in many systems, the sequence is what is called as KDEL. KDEL. They are single letter codes for amino acids. So, when you have a sequence called sequence KDEL, then that sequence is what is a ER retention signal. So, any protein that has a KDEL will not be packaged into vesicles and sent out, will be retained in the ER. Okay, so this is something that one has to keep in mind, that those proteins that are not folded properly, they are sent for degradation, and those proteins that are formed properly, they are either retained in the ER or they are sent out of the ER to further destinations. So, endoplasmic reticulum associated protein degradation is an inherent activity of the endoplasmic reticulum. So, ERAD factors are responsible for ensuring that the unfolded proteins are degraded outside in proteasomes. And therefore, one can say that the endoplasmic reticulum carries out quality control of the proteins. What do we mean by quality? Quality means that it functions optimally, it functions its best. An unfolded protein cannot function. It will not have a biological activity, while a properly folded protein will have a biological activity. So if it is not folded properly, first the ER ensures that because of the presence of molecular chaperones, it folds properly. But if it is still not able to fold the protein properly, then it ensures that the protein does not move forward because this protein does not have a proper biological activity. We also know that the rough endoplasmic reticulum forms the integral membrane proteins. So it is formed almost in the same manner. So the ribosome comes and sits on the translocon, and due to this signal peptide, it is lodged in the translocon, and translocon, you know, is the part of the membrane itself. So this stretch of signal start transfer sequence is a stretch of hydrophobic amino acids, which is why this stretch is able to interact with the bilipid layer. Please keep that in mind. It is able to bind to the bilipid layer because it is a stretch of amino acids that is hydrophobic. But those in these parts that are green, they are hydrophilic. So they will be pushed inside the ER lumen. However, there are many proteins that can have another stretch of amino acids that are hydrophobic. Okay, so then what happens is that this stretch, when it reaches the translocon, will again get embedded within the hydrophobic bilayer. So therefore, what happens is that that signal will remain bound. You see the stop transfer sequence will remain bound, so no longer will the protein go inside, but whatever has gone inside becomes the cytoplasmic domain. Whatever remains outside becomes the extracellular domain of the transmembrane protein. And this, as you can see, is a single pass transmembrane protein. The signal peptide is of course released by signal peptidase. So that is removed. Its job only was to ensure that it is translocating inside. The protein is translocating inside. Once its job is done, it is removed by signal peptidase. But there is no enzyme that is going to cut out the other series of amino acids that is hydrophobic. So that will remain embedded within the bilipid layer, and that's how you have an integral membrane protein. So both the stop and the start transfer sequence have amino acids that are hydrophobic, and therefore they lodge into the ER membrane. The sequence, if it has ER retention signal, then the membrane protein will remain in the endoplasmic reticulum. That way you would have, okay, now what you observe over here is that the C terminal end is outside and the N terminal is present inside. But you can have different ways by which you can have the single pass transmembrane protein. So depending on the hydrophobicity or the hydropathic region, you can have the N terminus outside and the C terminus inside, or you can have both the N terminus and the C terminus outside, but you have a part inside. Anything is possible. So depending on the sequence that is hydrophobic, you would have them arranged in a particular way. Now, this was single pass transmembrane protein, but you can also have multi-pass transmembrane protein. So in this case, you can see how you have two hydropathic regions, okay, two regions which are hydrophobic amino acids. They remain bound to the bilipid layer, but those regions which are hydrophilic either will form the intracellular domain or will form the extracellular domain. So therefore, you now have a bypass transmembrane protein. Similarly, depending on how many hydrophobic sequences you have, you can have a multi-pass transmembrane protein. So this you can see is the 7 transmembrane pass protein. So these, and in this case you can see the NH2 is inside, the COH is outside. Here you can see that both COH and NH2 is outside, but you can also have the NH2 inside and the COH outside. So all these arrangements is basically trying to tell you that integral membrane proteins are being formed in the endoplasmic reticulum itself. Now, another important function that the rough endoplasmic reticulum has is in a post-translational modification that is called as glycosylation. That means sugars can be added to the protein to form what is called as the glycoprotein. So the formation of glycoproteins can begin in the endoplasmic reticulum, but what is very typical is that the glycosylation in the endoplasmic reticulum is what is called as N-glycosylation. N-glycosylation. Why? Because the sugar moiety, so as you can see over here, this is an oligosaccharide moiety, and this oligosaccharide moiety that is added in the rough endoplasmic reticulum is same to all proteins. It is the same. And what is this complex? It is 2 molecules of N-acetyl glucosamine bound to first 3 molecules of mannose, then to another 6 molecules of mannose. So there are 9 molecules of mannose, and to 2 mannose, there are 3 glucose present. So this is the oligosaccharide moiety that can be transferred to a protein. So you see this is a protein. But the question is, where is this oligosaccharide moiety transferred to? So this oligosaccharide moiety is transferred specifically to an asparagine residue. So asparagine has a CONH2 at the end. So it is at the NH group that the sugar is transferred, and so it is called as N-linked glycosylation. So therefore, specifically the enzyme that is present in the membrane of the rough endoplasmic reticulum is only able to carry out N-linked glycosylation. So if this is the if this is the oligosaccharide transferase, this is the enzyme, then this enzyme is able to transfer a oligosaccharide moiety that is linked to the ER membrane. This oligosaccharide moiety is transferred to specifically an asparagine residue on a protein that has been translocated to the ER lumen. So this glycosylation is something that happens inside the ER. Now let us look at how this oligosaccharide moiety itself is formed. Interestingly, the ER membrane has something that is called as dolicol phosphate. This is of course a lipid moiety. Okay, and this dolicol phosphate is the phosphate to which you have the first oligosaccharide added, so that is N-acetyl glucosamine. So that's the first sugar that is added. Then a second sugar is added, and then there are 5 mannoses that are added. So two N-acetyl glucosamine and 5 mannoses are added outside, that is in the cytoplasm phase of the ER. So these are done or carried out by glycosyl transferases. So there are enzymes that carry out this sequential process of adding sugars. So you will always find the first one is always an N-acetyl glucosamine, the second one is always an N-acetyl glucosamine, and then there are 5 mannoses being added. Now, when you have this assembled oligosaccharide, then there is a flippase that is present in the ER membrane that flips this inside. So now the sugar moiety is present inside the ER lumen. Once it is present inside the ER lumen, there are more enzymes present inside the ER lumen that will add 4 more mannoses, and after having added the 4 more mannoses, 3 more glucose is added to form this final oligosaccharide unit. And as a whole, this oligosaccharide unit is transferred to a protein. So any protein that is glycosylated will always be glycosylated by this oligosaccharide moiety. Please to understand that once this oligosaccharide has been transferred to the protein, then This can be trimmed, this can be changed. That is, one glucose can be removed or one mannose can be removed. So you can have changes in the sugar moiety happening, but it is only after the transfer. So the presence of the oligosaccharide moiety, this is important, can also influence protein folding. Because we know that the oligosaccharide is hydrophilic, so therefore depending on how it is interacting with the surrounding aqueous environment, you would have the protein folding accordingly. What is also interesting to notice that GPI anchored proteins are also formed in the rough endoplasmic reticulum. So as you can see over here, this is a glycosyl phosphatidylinositol, and you can have a entire protein getting transferred to the sugar moiety of the glycosyl phosphatidylinositol to form what is called as GPI anchored protein. So you know many peripheral proteins are associated with the membrane through GPI anchors, and so you can understand that ER also is responsible for adding peripheral proteins to the membranes. Then of course it shares a function with the smooth endoplasmic reticulum, and that is that it is able to synthesize many phospholipids. So you can see over here how you have the phosphatidylcholine being formed. This is something that is carried out by the smooth endoplasmic reticulum also. So in fact, you must understand that even the membrane lipids are that may be going finally to a plasma membrane, or that may be going to a lysosome, or that may be going to a Golgi, is actually formed first in the endoplasmic reticulum, because these then form vesicles, and as part of vesicles they get exocytosed from here, but they get endocytosed at another place. So ER membrane, you can see how new phospholipids are added, then they are scrambled to form the symmetric growth of both the. So basically what we are trying to say is that new membranes can be, or more of phospholipids are formed or added to the membrane by the ER itself. As you can see over here, this that can be carried from the ER by exocytosis. So from the ER membrane, it can be exocytosed, and it can be fused to the with the plasma membrane. So now the plasma membrane will have these constituents, and then with the help of lipases, it can of course rearrange the entire lipid layer as it wants. So again, you can see how the plasma membrane can get new phospholipids from the ER itself. So you can see how even the lipids are being transported from the ER to the plasma membrane. So being part of the endomembrane network, this to and fro transport of not just the lipids but also the proteins is something that the ER is actively engaged in. So let us make the conclusions. The rough endoplasmic reticulum has ribosomes that help synthesize proteins that are to be secreted or are to be part of the endomembrane network system. As a protein is translocated into the lumen of the RER, the RER glycosylates protein and also does a quality check of the folded proteins. The membrane proteins, both single pass and multipass proteins, are formed in the RER, and other than those that are to be retained in the ER, the other membrane proteins are transported to other organelles and plasma membrane. The membrane lipids are also assembled in the rough endoplasmic reticulum and smooth endoplasmic reticulum to be distributed to different organelles. So the rough endoplasmic reticulum is a boon to the cell as it engages in shaping up of proteins that are distributed to different destinations, along with replenishing different lipids in lipid bilayers of the cell. It is integral to the endomembrane network system. Thank you.